Hello, my name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce Tracy Mullen, the Chief Operating Officer for Abex Biologics. Tracy is a chemical biological engineer from MIT who began her career as an antibody discovery scientist at a startup contract research organization specializing in vivo antibody discovery. In 2013, she joined the antibody discovery team at Biogen, where she helped implement the technology transfer of the Adimab yeast display platform for antibody discovery and engineering. Throughout her tenure at Biogen, Tracy worked to optimize the in vitro discovery of antibodies to tough targets, including leveraging the Adimab platform to generate antibodies against GPCRs. In June of 2018, Tracy joined Abveris Antibody as the Director of Antibody Discovery Operations, where she managed project design and development for in vivo discovery projects. She is now the Chief Operating Officer at Abex Biologics, formerly Abveris, a premium antibody discovery service partner boasting industry-leading timelines and cutting-edge technologies. With that, I turn this presentation over to Tracy Mullen to present her webinar titled High-Throughput Antibody Discovery Targeting Glycoproteins for Apoptosis Induction in Cancer. Thank you.
Great. Thank you for that introduction. I would like to start today by thanking both Wiley and Sartorius for inviting me to participate in this webinar, and also for providing Abveris the opportunity to present a case study from one of our recent antibody discovery campaigns. Because most of our projects at Abveris generally tend to involve blinded targets, we actually don't often get the opportunity to walk through case studies like this. So I'd also like to take this opportunity to thank our partner for allowing us to present this project externally. This is very exciting for us. Today's webinar centers around therapeutic antibody drug discovery for oncology indications. In particular, I plan to present a Hybridoma-Based antibody discovery campaign targeting a glycoprotein for apoptosis induction. I will start by providing a very brief overview of antibody therapeutics within the oncology space. I'll run through some background information related to this particular antibody discovery program, namely the high-level concept behind antibody-mediated apoptosis induction. And then I'll detail the work performed at Abveris, highlighting some of the high-content screening that helped aid in the discovery process. Finally, I'll end with some concluding remarks, and I'll also provide a very brief overview of our company for those of you interested in learning a little bit more. So with that, let's jump right into the orienting background information. Over the last 10 to 20 years, monoclonal antibodies have really emerged as some of the most successful therapeutic strategies in oncology indications, primarily thanks to their ability to directly target particular cell surface proteins on cancer cells. Upon binding to their particular target with exquisite specificity, antibodies may exhibit therapeutic activity through a variety of mechanisms of action, including, but of course not limited to, complement activation, neutralization of cancer-promoting growth factors, and of course, apoptosis induction. The earliest chemotherapeutic monoclonal antibody drug, rituximab, was first approved in 1997 for use in various types of cancers, including non-Hodgkin's lymphoma and leukemia. This particular drug was a chimeric monoclonal antibody targeting CD20, a protein that's found on the surface of immune B cells that, upon binding with this antibody, actually triggers cell death. Since then, a number of additional unconjugated monoclonal antibodies have been approved by the FDA for cancer therapy, including nivolumab, which I've actually pictured on the slide here on the left. Nivolumab targets PD-1, which is, of course, a very familiar immune checkpoint inhibitor, which a number of additional clinical candidates also aim to target as well. In fact, there's approximately 120 antibody-based immune checkpoint modulators currently in clinical studies, targeting immune system checkpoints such as PD-1, PD-L1, CTLA-4, and CD40, among others. Aside from unconjugated or naked IgG molecules, conjugated or armed IgG molecules capitalize on the benefit of monoclonal antibody specificity to provide a targeted attack to cancer cells. These drugs, commonly referred to as antibody-drug conjugates, typically involve antibodies utilized as transport vehicles that can deliver payload molecules such as cytotoxic drugs, cytokines, and even radionuclides to the exact tumor microenvironment. Technically, I guess bispecifics would also fall under the category of armed IgG molecules, so perhaps they should probably be in a category of their own. But either way, I think it's very safe to say that monoclonal antibodies in all of their various formats have truly revolutionized the cancer therapy industry. In fact, a quick review of the monoclonal antibody therapeutics currently in late-stage studies revealed a total of 33 cancer-related mAbs compared to 29 non-cancer-related mAbs currently in phase 3 studies. If you then break that out to include earlier stage studies, you'll notice a consistent trend of at least about half of clinical antibodies belonging to cancer indications. It has actually been estimated that monoclonal antibody therapeutics are granted FDA approval approximately twice as often as small molecule drugs, which perhaps helps explain the large number of mAbs in oncology-related clinical trials at the moment. That being said, monoclonal antibodies for cancer indications are actually less likely to be approved than those for non-cancer indications, potentially suggesting that improved development could be required for these particular drugs. Being able to test for biological characteristics such as specificity and function, for example, early on in the discovery process, could potentially help facilitate improved discovery of lead candidates and ultimately, hopefully, better clinical candidates. So to that end, let's review the basis of antibody-mediated apoptosis induction and see how an early apoptosis assay can be applied early on in the antibody discovery triage process to help identify lead candidates with functional relevance early on. To be effective as therapeutic drugs within the oncology space, antibodies, regardless of their format, must be capable of contributing to the elimination of tumor cells. Therefore, a number of therapeutics really capitalize on antibody-mediated apoptosis induction as a method of removing these cells. Not surprisingly, there are a number of mechanisms by which antibodies can induce apoptosis, some of which I've outlined on the slide here in the figure to the left. These strategies can involve direct targeting of cell surface antigens, receptor blocking, growth factor signaling modulation, ADCC, or, as discussed earlier, targeted delivery of cytotoxic agents. But regardless of the mechanism of action, being able to identify antibodies capable of kickstarting this process earlier on in the discovery workflow is really necessary for successful novel antibody discovery within the oncology field. And so, armed with this information, Abveris, together with a partner, designed an antibody discovery campaign to generate lead antibody candidates capable of inducing apoptosis through direct targeting of the cell surface glycoprotein that's known to induce apoptosis. The project design behind this discovery campaign centered largely around three main antibody therapeutic goals. One, cross-reactivity to both the human and cynomolgus orthologs of the target. Two, on-cell binding, and three, of course, apoptosis induction. Cross-reactivity to the murine ortholog of the target was considered a bonus, although likely a challenge due to the low homology across species. A hybridoma-based approach was leveraged for this particular program, capitalizing on the use of the unique genetically engineered mouse model specific to Abveris called the Diversimab mouse. Among other benefits, the Diversimab mouse has been engineered for increased diversity as measured by broader epitopic antigen coverage. We like to use the diagram pictured on the bottom left here to help really drive home this point. So with our Diversimab mice, which also happen to be somewhat autoimmune due to broken tolerance, we're able to access almost all epitopes present on the antigen of interest, regardless of homology to mouse. In fact, when compared head-to-head with wild-type mice, our Diversimab mice consistently deliver more unique antibodies with broader epitopic coverage than the wild-type mice. So you can start to really fully understand the power of this mouse model when you start to think about targets where, say, for example, the functional epitope of interest, so a ligand binding site, happens to exhibit poor immunogenicity in mice due to the high homology across species. So with this particular target in a wild-type mouse, you'd be very hard-pressed to pull out any antibodies that could bind to this region, but the Diversimab mouse will deliver. Since this particular target was a challenging one from a biological perspective, and since we knew that we were likely targeting a very narrow epitope if we did want that murine cross-reactivity bonus profile, we decided to leverage the Diversimab mouse for immunizations and antibody generation. An overview of the Hybridoma-Based antibody discovery workflow is presented on the right of the slide here. In short, This process involves diverse mass immunizations, titer testing, hybridoma fusion, high-throughput screening, and high-content flow cytometry screening for functional analysis. I'll plan to detail each of these steps in the subsequent slides, and then I'll conclude with a summary of next steps for this particular antibody campaign. Our immunization strategy for this campaign involved immunizing 3 diversimab mice using a recombinant extracellular domain version of the target of interest. Because we were looking for higher affinity antibodies, we opted to follow an extended immunization schedule to really capitalize on the in vivo affinity maturation process in these animals. After 8 weeks of weekly injections, a titer test was performed using sera collected from all 3 mice. We then ran the titer test in both high-throughput ELISA and high-throughput cytometry formats to assess the immune response generated in these animals. On this slide, I've included some data generated from the high-throughput flow cytometry analysis using the Intellicyt iQue flow cytometer. So for those of you not familiar with this instrument, the iQue actually allows us to analyze an entire 96-well plate in about 8 minutes in up to 6 different detection channels. We can then view in real time what Intellicyt refers to as actionable data or live results as they're collected on the instrument as a function of time. So you have a particularly anxious project leader, for example, who really wants to see the results as soon as possible. This would be the fielded view that you'd show them in real time while the plate is actually running on the instrument. On this slide, you'll see an example of this type of real-time analysis where we're looking at antibody binding on the y-axis as detected using an anti-mouse ELISA-488 conjugated secondary as a function of runtime on the x-axis. The serum itself is titrated, starting at a 1:100 dilution and diluted 3-fold out 11 points across the plate. As a result, you can see profiles like those shown on the slide here with decreasing signal intensity over time. These samples were run in duplicate, so 2 titration series per mouse, and the plate was analyzed in row format. So what you're looking at here in these 4 different charts are the 3 mice that we immunized with the target of interest, and then naive mouse serum that's run as a negative control. An alternative way to view this data is in a heat map format, which the iQue software also really easily generates after data acquisition is complete. In this format, you can see the ROT and the phi values overlaid on the heat map, and these ROT values can actually be exported directly to a CSV file if you'd like to analyze this elsewhere. Alternatively, we can also plot these MFIs, again using the iQue software, to generate graphical representations of the titer results with error bars to help understand data consistency across replicates. I've overlaid the corresponding graphs for each mouse on this slide here for those of you more interested in seeing titer data in this format. But regardless of the final format for data representation, the iQue really helps enable quick decision-making to help progress antibody discovery workflows involving on-cell screening. As you can see on this slide, we were able to very quickly determine that all 3 mice exhibited titer against this particular target of interest, which meant we were ready to proceed to fusion with these animals. Spleen and lymph tissue were harvested from all 3 mice. Collected B cells were fused to a partner myeloma cell line, and subsequent hybridoma products were plated out across 30 96-well plates for downstream analysis. Given the availability of the human and the cynomolgus recombinant protein reagents, and also given the lack of cyno target-expressing cell line availability, we opted to perform a primary fusion screen using high-throughput ELISA instead of high-throughput flow cytometry, and pulled forward clones that found both the human and or the cyno recombinant protein with an ELISA OD of 0.5 or higher. So you'll see on the slide here just a representation of what that data looked like, with clones in the red in the bottom left there being clones that we ended up not pulling forward, and anything in green being hits that were carried forward to a secondary screen. So we then carried all of the interesting hits from the primary screen to the secondary ELISA screen, in which we also incorporated a murine recombinant protein. This time, we set our OD ELISA threshold at 0.4 or higher, and again pulled forward any clones that found the human and or cyno recombinant protein. I've also highlighted on the slide here in red with arrows 4 clones that exhibited murine cross-reactivity at this secondary screen stage, which of course we were very excited to see given the lack of murine recombinant protein incorporation into the primary screening workflow. As a reminder, murine cross-reactivity was considered to be a bonus goal for this particular campaign, and with an overall homology of around 60% to the human protein that we used for immunizations, This cross-reactivity was actually considered to be somewhat of a lofty goal. So for these reasons, among others, it was very exciting to see some candidates exhibit this triple species cross-reactive binding profile. All in all, the high-throughput ELISA primary screen helped us identify 30 parental hybridomas of interest, 50% of which appeared to be human and cyno cross-reactive. All 30 parental hybridomas were expanded for additional subsequent ELISA-based analysis, in which 14 parental hybridomas of interest were selected to be carried forward as clones of interest. These 14 parental hybridomas exhibited varying cross-reactivity profiles that I've outlined on the slide here in this summary, but most importantly, 4 of these clones exhibited that triple cross-reactive profile. Alongside our ELISA-based secondary screen, we also ran a flow-based secondary screen on the 30 parental hybridomas of interest that were carried forward from the primary screen. These clones were analyzed for cell surface binding on a cell line that's expressing the human target of interest, so in other words, that same cell line that we used for the titer testing. Interestingly, most ELISA-positive hits did not exhibit on-cell binding, and in fact, there did not appear to be any significant correlation between ELISA activity and flow activity. While this was, of course, an unfortunate outcome for our secondary flow screening, it really helps highlight a very important lesson in antibody discovery. Recombinant proteins might not always be an accurate representation of the corresponding cell surface target. Therefore, it is imperative to fully validate a recombinant protein ahead of immunizations and/or campaign screening to ensure that you're generating and screening for antibodies against the relevant format of the antigen of interest. Additionally, these data help highlight the importance of high-throughput screening, especially on-cell screening, as early as possible in the final relevant format. So in other words, if you're looking for on-cell binding at the end of the day, it makes sense to screen on cells early on in your triage process. For that reason, Abveris aims to incorporate high-throughput flow cytometry using the iQue platform as early as possible. Unfortunately, as a reminder, we were unable to do that in this particular campaign due to the lack of availability of a cell line expressing the cyno target of interest. And again, as a reminder, cyno and human cross-reactivity was a requirement for this particular campaign. That being said, we do aim to follow that screening strategy for most of our other antibody discovery campaigns that involve targeting of cell surface proteins. So, while we were able to identify 14 clones of interest from the ELISA-based secondary screen, we were really only able to identify 5 clones of interest that showed any appreciable flow binding to carry forward to the next steps of the campaign. That next phase involved high-content iQue screening for functional characterization of our lead candidates. So this is where the apoptosis assay came into play. On the right side of the slide here, I've included a very brief overview of the key steps involved in this apoptosis assay. Basically, our workflow involves treating the target-expressing cell line with a purified IgG sample at a set concentration of 20 nanomolar for all 5 IgG samples. We incubated these cells in that IgG sample for 20 hours at 37 C, and then we stained the cells with a live dead marker and an annexin 50 conjugate to identify dead cells and cells actively undergoing apoptosis, respectively. We also incorporated camptothecin into this assay. This is a small molecule topoisomerase 1 inhibitor known to induce apoptosis, with which we treated cells for 4 hours prior to staining to act as a positive control. This assay was set up as a single-point analysis with regards to both antibody concentration and incubation time, and samples were run in triplicate. The plate setup for this assay can be found on the top left of the slide, and what you're really looking at here is a plate-based view of corresponding iQue-generated results on the bottom left of this slide as well. This plate-based view is essentially a dot plot summarizing the 2 key readouts from this experiment. On the y-axis, you're looking at the annexin 50 staining, and on the x-axis, we have the live dead staining. So therefore, any overall population shift upwards indicates cells undergoing apoptosis, and any overall population shift to the right indicates dead cells. A summary of the results can be found on the bottom right figure of this slide, where you can see 3 representative clone readouts, an isotype control and camptothecin results. 2 clones were actually left out of this summary due to their similarity to clone 2 in terms of lack of apoptotic activity. But as I'm sure you can appreciate, clone 5 here really stands out among the others with a clear increase in percentage of dead and apoptotic cells over even the camptothecin positive control. You'll see a note here above this particular clone, which reads, underestimated, nearly all cells disappeared. This is also highlighted visually in the dot plots for this particular clone on the plate-based summary results that I've also highlighted here in yellow. So in order to better understand what we mean by this, I thought it might be prudent to very briefly go over how the iQue collects events and presents results. So unlike your typical flow cytometer where you set a particular number of events that you're interested in interrogating and then collect until you reach this mark, the iQue actually operates by a set SIP time in which it pulls a certain volume of sample for every single sample on the plate for that predetermined SIP time per well. Therefore, assuming that you plate the same number of cells per well, you would, in theory, anticipate a similar number of events to come off of the instrument for each well, assuming, of course, consistent sample treatment throughout the staining process. In this particular assay, we aliquoted a set number of cells in each well and treated each with its corresponding IgG sample. The following day, the samples were handled together in aggregate using a high-throughput liquid handler to avoid any operator bias being applied to any particular well. The plate was then imaged using a set SIP time for each well, which was the same for all wells, and a similar number of events were actually collected for all samples, except, of course, for the 3 wells that were incubated with clone 5. Interestingly, all 3 clone 5 wells showed a significant decrease in the number of events present for iQue analysis, suggesting, perhaps, that the cells may have progressed through apoptosis and cell death over the course of the 20-hour incubation. Additional analysis, including multiple concentrations and incubation times, are of course required to further elucidate the cause of these observations, but the overall increase in annexin 50 in live dead staining certainly suggests that this clone is worth further investigation. In addition to apoptosis analysis, we took advantage of the high-throughput nature of the iQue screening tool to determine on-cell apparent KD for each of our top 5 candidates of interest. Shown on this slide here is one of our top 5 clones analyzed for on-cell KD determination using the iQue dose response analysis functionality. The assay setup for this workflow is fairly straightforward. So essentially, we perform a titration series of known antibody concentrations and then plate that and put that onto the iQue software and allow the iQue software to collect events and perform a nonlinear regression to determine that EC50 measurement for each response curve. So in a matter of seconds, really, the software outputs the estimated EC50 value, which, as shown here for this particular clone, came back at 1.39 nanomolar. Conveniently, the estimated affinity for the same particular clone as measured by Octet was actually quite similar to that measured via on-cell apparent KD analysis. These results are actually shown on the bottom of the slide here, where you can see the titration series performed for this same particular clone against the human recombinant protein on Octet. Unfortunately, though not surprisingly, not all clones showed comparable KD measurements by both Octet and on-cell apparent KD, though this is to be expected given the inconsistencies observed between the recombinant protein and the cell surface antigen as discussed earlier in the presentation. So in fact, this particular clone highlighted on this slide was really the only clone that showed both strong flow activity and decent ELISA binding at the secondary screen stage. So I guess it's not too surprising that this clone would provide comparable results across both assay formats. The remaining clones were also analyzed via on-cell apparent KD, and the resulting titration curves can be found on the right of the slide here. Beneath this graph, I also included the actual KD measurements that were returned by the iQue software with varying degrees of reliability. So as you can see, 2 of the top 5 clones here that were analyzed appeared to exhibit very poor affinities with non-saturating profiles observed for both titrations, even at the most concentrated dilution point of 1 micromolar. Therefore, the estimated on-cell apparent KDs are likely unreliable for these 2 clones, but the remaining 3 clones exhibit a variety of reliable affinity measurements. So what have we learned here and what's next? As discussed early in the webinar, it's clear that monoclonal antibodies continue to dominate the clinical landscape for oncology, and that antibody-mediated apoptosis has proven to be an effective method of cell killing. In order to identify the most promising antibody candidates early on in the discovery process, it's really important to incorporate high-throughput screening in relevant downstream formats as early as possible. And high-throughput flow screening is really an exceptionally convenient and beneficial way to handle this triage process. Through the use of the iQue flow cytometer, Abveris was able to successfully identify a lead therapeutic candidate capable of exhibiting apoptosis induction and single-digit nanomolar affinity to the target of interest. Currently, Abveris is working on further analyzing this lead candidate, including repeating the apoptosis assay with a dilution series of the antibody of interest alongside some benchmark antibodies for comparison. Assuming promising results, the next step for this antibody would be humanization and in vivo studies. So to conclude this webinar, I thought I'd just provide a very brief overview of Abveris and Avex Biologics for those of you interested in learning more about our two companies. So Abveris is a contract research organization specializing in critical reagent antibody discovery. We offer a sequence-to-sequence workflow, meaning we can take an antibody discovery campaign from antigen expression through immunizations and discovery, and ultimately deliver antibody sequences and recombinantly expressed material if desired. We boast a large number of proprietary immunization strategies and adjuvant cocktails for tough target antibody discovery, and a suite of premium downstream characterization assays to really screen for lead candidates. Due to the combination of proprietary mouse model, immunization techniques, and screening processes, we specialize in high homology targets, constrained epitope targeting, and anti-idiotype antibody discovery. All workflows start with mirroring immunizations, and we offer both fully human and proprietary engineered mouse models for discovery purposes. Avex Biologics is our newly formed sister company specializing in therapeutic antibody drug discovery. The Avex platform for antibody discovery leverages both the upstream and downstream strengths that Abveris workflow offers, so namely the proprietary immunization and characterization expertise, but incorporates the Berkeley Lights Beacon platform for single B-cell screening and selection. With the Beacon platform, we're able to screen for desired antibody characteristics such as antigen specificity, species cross-reactivity, functionality, and even developability on the same day as tissue harvest, meaning the workflow offers a discovery timeline as rapid as 35 days from immunization start to target-specific sequence identification. This workflow, therefore, is ideal for anyone looking for rapid, high-throughput, high-resolution screening at the single B-cell level, utilizing the most rigorous plasma B-cell screening workflow in industry. And with that, I'd like to take any questions.
Thank you very much for that very informative webinar. Our first question is, have you run side-by-side epitope binding analysis for your Diversim AB mouse alongside a wild-type mouse as a function of immunization strategy? And if you have, do you see a loss of diversity with longer immunizations?
Yeah, so we actually have run an epitope binding case study in which we compared immunization strategies in our Diversim mice alongside Balch B mice. And we saw very interesting and somewhat surprising results, at least according to the rules of immunology. So in this head-to-head comparison, we basically immunized two cohorts of Diversim mice and two cohorts of Balch B mice with the same recombinant protein antigen, and those two cohorts would follow either an extended or accelerated immunization strategy. We then performed fusions, carried forward antigen-specific hits through primary and secondary screening to ultimately isolate a panel of 120-ish antibodies that we then used in combination with our in-house CAR-TERA platform to screen for high-throughput binning. So in this experiment, we essentially binned all 120 antibodies against each other. And the results of this binning experiment actually indicated that the antibodies coming out of our Diversim mice covered more epitopes than those from the Balch B mice. So basically, we saw antibodies from our Diversim mice binning with all of those from the Balch B, and then some additional epitopes even outside of those that the Balch B mice antibodies fall within, but only the Diversim antibodies were found. So if you actually break out those Diversim antibodies into their original immunization cohorts, which we of course did as part of this analysis, we saw comparable epitopic spread across both cohorts of immunization. So really what this can be boiled down to is that our Diversim mice definitely seem to maintain epitopic diversity regardless of immunization duration.
Thank you. So regarding some of your top clones, why do you suspect that one of your top clones
binds equally well to both recombinant protein in cells but not the others. If the recombinant protein is a bad representation of the cell surface protein, wouldn't you expect all antibodies to show only ELISA binding?
Yeah, this is a really great question and one that I fully anticipated receiving when putting together this presentation. So in short, I suspect there are likely a number of factors at play here, but the most likely scenario, at least in my mind, is related to the overall recombinant protein integrity. So if this protein, for example, was completely misfolded and or aggregated or degraded, I would have expected zero correlation between recombinant protein binding and cell surface binding. And sometimes with some projects we do see stuff like that occurring. But that being said, it's entirely possible that the recombinant protein prep in this case was not completely garbage. So perhaps some epitopes were appropriately presented on the recombinant protein, thus allowing for antibodies that recognize these epitopes to bind both the recombinant protein and the cell surface target. There's of course other additional potential explanations as well. I mean, perhaps there were some post-translational modification differences between the recombinant protein and the cell surface protein. For example, it's really hard to say without knowing more about exactly how the recombinant protein was made. But as mentioned briefly in the webinar, I think in general, the best strategy to avoid issues like this would be to screen in the appropriate end-use format upfront.
Thank you. How do you know that the method of killing was not Fc mediated? Did you reformat your antibodies into effectorless molecules?
We did not reformat our antibodies into effectorless molecules, no. Without giving away too much information, I can say that the cell line that we were screening on was a human line that did not express any Fc receptors.
Thank you. So for a customer who wants to use a recombinant protein as an immunogen, but requires on-cell binding, do you have any suggestions for them about how to go forward?
Great question. So we frequently run campaigns where partners provide a nicely validated recombinant protein, and then we'll use that for immunizations and we'll screen on cells. And we've actually seen kind of a mix of both really successful campaigns where the protein that's provided is a nice representation, and then some not so successful campaigns. And I think that really depends on whether or not, of course, this protein is reflective of the cell surface target and whether or not it's been nicely validated up front. So in general, whenever possible, we prefer that our partners validate the recombinant protein, perhaps with positive control antibodies or its natural ligand binding partner, something like that. And preferably confirming biological activity up front. I can say that we can usually tell at the titer test stage whether a recombinant protein is an accurate reflection of the cell surface target, because we'll usually run a side-by-side ELISA and flow titer test. But at that point, it's kind of a little bit too late, right? You've already immunized the animal. So really validating that protein up front is imperative.
What other types of functional screening can you do using iQue?
So we actually work with the iQue Screener Plus, which is a 6-channel flow cytometer. So really, any functional screen that requires 6 or fewer readouts can be applied to the iQue. At Averis, the most common functional screens that we run using our iQue are apoptosis assays, neutralization assays, and sometimes ligand blocking assays. But we've more recently been looking to optimize additional screens, such as antibody-mediated receptor internalization, for example. We have a very enthusiastic flow cytometry team here, always interested in running new screens. So if whoever asked this question has a functional screen that they're looking to outsource, please reach out after this webinar.
You mentioned a hyperimmune mouse model that worked well for this target. Could you just tell me a little bit more about that mouse model and what other types of targets it is applicable to?
Yeah, sure. So really there aren't too many target classes out there for which we can't leverage this diversimab hyperimmune mouse model. Of course, with some obvious exceptions like toxic immunogens and things like that. But due to the hyperimmune and autoimmune nature of our mouse model, and paired with some of our proprietary immunization strategies and adjuvant cocktails, we really specialize in high homology targets, surrogate antibody discovery, constrained epitope targeting, really any traditionally difficult and or non-immunogenic targets, so antibody idiotypes, for example, or small molecules, and of course, complex cell surface targets that require specialized immunization techniques. We definitely have a pretty impressive vivarium that loves to tackle challenging projects, so we kind of get those types of projects pretty frequently, and it's a lot of fun for us to work on those.
So are there timeline advantages to using the hyperimmune model system?
Absolutely, yeah. So we have a variety of, I guess you could say we have a very unique mouse model, right? So in working with our hyperimmune mouse model, we can actually run a number of different immunization strategies in terms of length as well. And so we can leverage our diversimab mice in accelerated immunization campaigns, which will likely result in a panel of antibodies with significant diversity in both epitope coverage and affinity in that industry-leading timeline. So the accelerated immunization protocol is basically 3 weeks long, and we couple that with a streamlined, very high-throughput downstream characterization workflow that keeps the discovery timeline as competitively short as possible. And that's with regards to our hybridoma workflow. So alternatively, I briefly mentioned our beacon workflow. This is actually even faster. So screening results can be read out same day as tissue harvest. So we'll be able to know that day whether we have any hits of interest, which shortens the discovery timeline from months to even days.
So what are some other immunization strategies you can use instead of the recombinant proteins, especially in the case where you can't either make one or you don't have one available?
Yeah, so there are certainly target classes out there for which it's really challenging to obtain a recombinant protein, right? So if you think about ion channels, GPCRs, for example, it's really challenging to develop a reliable recombinant protein. So in those cases, we follow one of 3 immunization approaches depending on reagent availability. So we'll either follow a DNA immunization, whole cell immunizations, or subtractive immunizations. And I think the first 2 are fairly straightforward. The last might be unfamiliar to some audience members out there, so I'll go into that a little bit. So basically, we leverage our subtractive immunization strategy in situations where partners only have a single cell line available for both immunizations and screening, and they want to minimize background binding to the host cell endogenous surface protein. So in this case, we'll leverage our subtractive immunization program. We'll tolerize the mice before immunization using an untransfected or preferably a mock transfected host cell line. And we accomplish this by basically injecting our animals with the toleragen cell line and then subsequently treating them with an immunosuppressive drug that obliterates the immune response against that cell line. We'll then follow up that tolerization period with an immunization period, this time using the same cell line that you had used originally, but this time expressing the target of interest. So in this case, the only sort of novel target present to the immune system is that antigen of interest, thus resulting in more of a targeted response against that antigen. And we actually have some really nice case studies detailing this procedure. So again, follow up after if you're interested in learning more.
Thank you. So one question that somebody sent in is, why use an in vivo discovery platform instead of an in vitro-based one?
Oh, this question hits close to home. So I consider myself a discovery platform agnostic antibody discovery scientist, but I think it's fair to say that there are pros and cons to both in vivo and in vitro discovery platforms. So if you're looking to speak exclusively about the benefits of in vivo, I'd say probably the biggest benefit is the natural affinity maturation that happens in vivo. So when you're dealing with animal models, you get the added benefit of natural genetic recombination, followed by somatic hypermutation and clonal selection. So essentially just built-in AFMAT. And oftentimes antibodies coming out of in vitro discovery approaches require an extra few rounds of affinity maturation and potentially additional engineering for optimization. That being said, unless you're leveraging a single B-cell screening approach after immunizations, you tend to lose that genotype-phenotype linkage for directed evolution, and that's really the hallmark of in vitro discovery. So there's that trade-off there. So again, just coming back to how I started, there's really just pros and cons to both platforms.
Great. So say I'm just starting off on something like this, and I'm at the very base platform, what are some of the ideal reagents that I would need to prepare for an antibody discovery project?
So reagent quality is really everything when it comes to antibody discovery, and that's true for any platform. So when planning an antibody discovery campaign, I'd say the most important consideration would be your primary antigen of interest. So you want to ask yourself questions like, is this antigen the best quality possible? Is the antigen prep biologically and functionally relevant? Have you confirmed benchmark antibody or ligand binding activity? Sometimes and often, actually, counterscreening reagents can be just as important as the target reagent. So if, for example, you have a very small recombinant reagent with a large immunogenic tag, for example, it would be imperative to provide a counterscreening reagent with a similar tag to eliminate candidates that don't bind your target of interest. And at the end of the day, every good scientist loves a positive control. So wherever possible, if you can provide a positive control, that will greatly improve the chances of success, or at least consistency.
Well, great. Thank you for those answers. So we've reached the end of our time today for questions. I'd like to thank Tracy Mullen of Veris and Abex Biologics for taking the time to present to us her talk on high-throughput antibody discovery targeting. Any questions that were not addressed will be answered by email. And please feel free to reach out with any additional questions. This presentation was sponsored by Sartorius in partnership with Wiley's Current Protocols. This webinar will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section. Thank you for attending.